Hello, everyone, and hope that you're doing well. Now, today we're doing another review of a CSEC biology paper. This is a paper 2 from May, June 2018. Now, please remember before you start the examination that you should read through the instructions carefully. And if you should have any questions or queries, you clarify those with the examiner before the examination begins. Now, let's jump into section A, question number 1. And part A reads that scientists interested in the studying of the survival of organisms living at high temperatures identify some microorganisms living in a desert habitat where the average daily temperatures exceed 50 degrees Celsius. They extract an enzyme protease that facilitates the digestion of proteins from these organisms and investigate the effect of temperature on the action of this enzyme. So part 1 is to state 2 precautions that they should take in carrying out this experiment. And one of those precautions is to extract the enzyme at a temperature below 50 degrees Celsius. And this is really to prevent the denaturing of the enzyme. Another one could have been that to store the enzyme immediately to prevent it from oxidizing. And of course, if enzymes are oxidized, then their nature will also be changed. And another one could be to maintain a neutral pH environment. And that is to ensure that the enzyme, if it's sensitive to high or low pH, will not be affected. And also is to ensure, which is the fourth one, is to ensure to clean all apparatus. And of course, if you list any other suitable ones, then yes, those will be accepted as well. All right, so now for part 2, it said the results of the experiment are presented in table 1. On the grid provided in figure 1 on page 5, draw a graph to represent the results presented in table 1. And so the 2 things we are working here, the 2 variables, one is temperature and the other one is time taken for protein to disappear. And that is in minutes. So temperature is in degrees Celsius. Now, one observation here, if you notice that the increments between the temperature is not the same. So you notice it move from 5 to 10 and then from 10 to 30 and then from 30 to 35, right? So the incremental changes are not the same. So in this case, a line graph is best. All right, and so to draw this graph, so first let's point out a few things. One, when you're drawing graphs, you should always have a title because normally give a 1 mark for the title. Also, it's very important to label axes as well. And so I put time on the Y, time for the protein to disappear on the Y, because the disappearance of the protein is actually a dependent variable. And time, sorry, not time, but temperature actually is the independent variable, and that should be on your x-axis. All right, and so the graph will shape something like this, just to kind of scroll up and you'll see. So notice here it is very high and then it comes low. And I want to point out that for this graph, since we're talking about the time for disappearance, the lower the time, which means the faster the reaction is. All right, so don't be misled by the shape, okay? So the lower the graph is the faster the reaction because the time taken for the disappearance of the protein is low. All right, and so part 3 is that from the graph in figure 1, identify the optimum temperature for the protease activity. And a matter of fact, you can even use a chart to tell you this because it is 55 degrees Celsius. This is when you have the least amount of time taken for the disappearance of the protein. All right, and so for part 4 is to suggest one reason for the shape of the graph at less than 10 degrees Celsius and 2 reasons for the shape of the graph between 10 degrees Celsius and 55 degrees Celsius, and 1 reason for the shape of the graph at more than 55 degrees Celsius. And if you go back to the graph, you'd realize that less than 10 degrees Celsius, it was 60, which is meaning taking a very long time. That's a whole hour. All right, so 60 minutes, which is 1 hour. And so at that point, we can state that the enzymes are inactive. So the enzyme is definitely inactive at low temperatures. And from 10 to 55 degrees Celsius, what is happening here is that the enzyme become activated. And also the enzyme activity increase with increase in temperatures. All right. And also the point to make right here is that the enzyme reaches maximum activity at 55 degrees All right, and so this is the most suitable temperature, what we call the optimal temperature for the enzyme action or activity. All right, now for more than 55 degrees Celsius, then the enzyme is starting to denature. And if you notice, if I should go back to the graph or to the chart, you'd realize, let me just go back quickly. Here you can see that the time taken for the disappearance, so it's after 55, which is right here. So after that, the time for disappearing of the protein start to increase. So at that point, because the temperature is so high, the enzyme itself start to denature. All right, and so let's go into part 5. And for part 5, it says describe a suitable procedure that the scientists could use to investigate the effect of pH on the activity of the protease enzyme. And so the temperature, the outline of this experiment could have been: one is to add substances of varying pH levels to different test tubes. And this is to ensure, and first thing to ensure, this is a precaution as well, is to ensure the same volume of substances. All right, so you make sure you maintain the same volume. And also for the second point or step that you'll do is to add the same amount of the enzyme to each test tube for the same time period. All right, and so you want to do it at the same time, or if you do it differently, make sure you have to use the same time. And then the third thing to do is to add the same quantity of protein sample to each test tube. And then what you want to do after that is determine the time it takes for each protein sample to disappear. And then you want to record the result in an appropriate chart or table. All right, and so that's a simple outline of the experiment to test for pH. It's the same thing as doing temperature, but you only take away the temperature part and substitute that for pH. For part B, it's the human skin. In figure 2 is an organ which helps human to regulate their body temperature. And so they ask us here to name the parts, label 1, 2, 3, and 4. And so they have, I label them on the diagram as well. So 1 is the hair shaft, 2 is the sweat gland. They got a T out of this, so sweat gland. All right, and we have here now, we have the erector muscle cell. All right, and so this is erector muscle, or I can say the hair erector muscle. And now we have part 4, which is the capillary. All right, I can say blood capillary as well. All right, so those are the different parts on the skin. 4 easy marks. Now for part 2, we said describe how the structures named in part B1 helped humans to regulate their body temperature. And the hair shaft, it kind of worked in terms of laying down and standing up straight. And so when the body is hot, right, the hair lay to prevent the trapping of air. And when hair is trapped, it creates an insulation, just to make a note of that. And so when the body is cold now, the hair rise to trap air, creating an insulation. And for sweat gland produces sweat to cool the body, and this will lower the body temperature. So when you're hot, you produce sweat. And just to make a note, that when sweat evaporates, it takes away heat from the body. All right? Now, the erector muscle cell now, that is connected to the hair itself, it controls the movement of hair. And so when the body is hot, the erector muscle relaxes and hair will lay flat. All right, so kind of tie in with number 1 if you notice how the hair works. And then now when the body is cold, the erector muscle contracts and the hair will rise, and such you'll have goosebumps. And the rising of the hair is really to trap air to create an insulation around the skin. All right, so you maintain the body temperature. And for capillaries now, the capillaries now is that when the body is hot, the capillary will dilate, all right, and that is called vasodilation, and which means the capillaries get wider, and because of this, they'll be closer to the surface of the skin, and they will lose heat to the environment. And the opposite is that when the body is cold, the capillary constricts, and the constriction of the capillary is called vasoconstriction, and this is to retain heat. All right, and so let's slide down to the next question, which is part C. It's to explain how changes in the epidermal cells of leaves help plant to conserve water during high temperatures. And when I say the epidermal cells, I just want to point this out. You have the epidermis, right? And so any cell that is found within the epidermis are called epidermal cells. And so there are a number of different cells that are there. And so the one that we're going to focus here on that controls water or to conserve water is the guard cells. And so the guard cells within the epidermis, when the temperature is high, the guard cells themselves lose water, all right, and they become flaccid. And once the guard cells are flaccid now, they come together and close the stomata. All right, and the point to note, you notice the piece in red right there said when the stomata are closed, water is unable to evaporate. And so water is conserved as a result. All right, and so that's a 3 marks, and that's end question number 1. For question number 2, is a figure 3 shows 3 different types of cells, A, B, and C. All right, and I know that you know these cells by looking at them. All right, and so here's a name, the type of cell labeled A, B, and C. And so A is animal cell, B is a plant cell, C is a bacterium or cells that associated with bacteria. And then for part 2 is a name 2 structures that are common to the 3 types of cells. And so we have a number of things that are common to these cells. And we have here, cell membrane is common to all cells, in fact. All right. We also have DNA that is common to all the cells as well. And we also have ribosomes. All right. So these are the common structures that are for these cells. All right. All right. So let's now go into part 3 of this question. All right. Part 3 of this question, it states 2 differences between the type of cell labeled A and the type of cell labeled C. All right. And just as a reminder, A is the animal and C is the bacterium cell. All right. And so animal cells, no cell wall is present. For bacteria, yes, cell wall is present. For cell A, no pili, but pili are present in the bacterium cell. A point to note, right? Normally you see, or some bacteria, you see flagellum, right? You see flagellum. However, for that diagram up top, there is no flagellum shown, only pili. And just to make a note, the difference with pili and flagellum, they are similar, but the difference is that pili are shorter and thicker. Flagellum tend to be longer, all right? And so they also serve different functions as well. The pili is used to stick on surfaces, while the flagellum is used for movement. However, sometimes the bacterium can use the pili to move as well, but it generally is used to stick to surfaces. All right, in animal cell, there is no nucleus, right? However, in animal cell, there is nucleus, and there's no nucleus present in bacterium cell. All right, and point to note here is that in animal cells, which is a eukaryotic cell, the organelles are surrounded by membrane. So they are membrane-bound organelles. And so in the bacterium cell, which is a prokaryotic cell, there's no membrane-bound organelle. All right, and just to know the difference between prokaryotic and eukaryotic, that's pretty much what they ask you here. And once you know that, you're straight. All right, for part B, is the tissues made up of type B cells are placed in a container of concentrated salt solution after and left for 1 hour. And so again, this is a plant cell, right? Just to bear it in mind. It's to describe 3 changes in the appearance of the cells after 1 hour in the concentrated salt solution and give an explanation to account for these changes. And so here, the changes that will be there is that one, the cell will lose water. So that's one change that will take place there. And so what will move from the inside to the outside of the cell? That's pretty much what will happen. And so the cell membrane will move, the cell membrane moves away from the cell wall, and the vacuoles, they decrease in size or volume, and the cell tissue, the cell, which is the tissue, the cells together, which is a tissue, become flaccid or plasmolized. So all the cells will become flaccid or plasmolized. So the explanation is this now, is that water moves from an area of high concentration, which is inside of a cell, to an area of lower concentration, which is outside of a cell, across a plasma membrane or across the plasma membrane of the cells. All right, and that just describing osmosis, all right, because osmosis is the reason for this to take place. And the point to note that the solution there is hypertonic. all right, which is PER, hypertonic solution. All right, and so the next part of the question now, which is part C, said tissues made up of type A cells and type B cells are placed in a container of distilled water and left for 1 hour. So type A and type B, again, is two different cells. So the type A will be the animal cell, the type B will be the plant cell, and they place in distilled water. So describe how the appearance of type A cell is affected to differ from that of type B cells and give an explanation for the difference. Now, point to note here is that type A cells, which are animal cells, will increase in size until it bursts or till they burst. I have it here till let's change to they because since I say type A cells, so they will burst until they burst. All right, and so the word we use for bursting of the animal cell is lyse, they lyse, which means to split or to open. All right, however, now type B cells, which are plant cells, will remain or maintain their shape. So they maintain their shape. All right. And the explanation for this is that type A cell, which is animal cell, they do not have any cell wall. All right, so no cell wall is present to contain the increase in pressure. All right, and thus unable to prevent the cell from bursting. However, now for type B now, they have cell wall that is able to withstand and contain the increased pressure, thus maintaining the shape and preventing them from bursting. All right, so this, the difference in cell wall between these two cells will give a difference in result in terms of the effect of the distilled water. Now, for question number 3, it's a figure 4 shows a section through the human eye, and we label all the different parts. And so that's what they actually asked us here, I think. Right. And so he said, table 2 is incomplete. It says shows structures in the human eye labeled in figure 4 and their related functions. It's a completed table by matching each labeled part of the eye to its function. Write the letter and the name of the part. It said the first row has been completed for you. And so just to make a quick review of the diagram in terms of the different parts, you can just go through them. So we have the conjunctiva, which is A. B is the ciliary muscle. This part here, the two with the lines right here, that would be the suspensory ligament. I should have labeled that. We have the iris, we have the lens, we have the sclera, and we have the retina. And between the retina and the sclera, what we have is the choroid. All right, and then we have the optic nerve. And of course, right at this point here, we will have our blind spot. All right, and so now let's just slide on into the chart or the table. And it said now for the first row, conjunctiva is memory that protects the cornea. And so transmit nerve impulse to the brain, that is optic nerve, which is labeled D, and then becomes, the next row is it becomes more or less convex to focus light onto the retina. And once you see the word convex or become more convex, you know that is the lens for sure. All right, and the next one is to control the amount of light passing through the pupil, and that will be the iris. Contracts and relax to alter the shape of the lens, and that is the ciliary muscle. The suspensory ligament also plays a role in that as well, but is not labeled. So definitely we go with B. And then we have here, now, a contracts and relax to alter the shape. No, we'll just go through that. So that one is the ciliary muscle. The next one is to protect and keeps the shape of the eye, and that is the sclera. And the last one is to convert light energy to nerve impulse, and that is the retina. And remember that the retina contains rods and cones, all right, which are light sensitive cells. Now we said name one type of light sensitive cell found in the retina, and you can either write rods or cones. And for part C, it asks us to explain why colors are seen less clearly in dim light than in bright light. And a point to note is that rods are more sensitive in dim light. All right, and so rods are more active than cones in dim light conditions. And so point to note again is that rods do not sense colors. And so there's little to no colors are seen in dim light, since cones are less active, because cones are used to detect colors, or they are sensitive to colors. All right, and at that point, they are not active. They are slightly active, if any at all. All right, for part D, it's a color-blind woman meets a man who has normal color vision, determines the probability of this couple having a colorblind daughter by completing a genetic diagram below. And so they gave us the parent here, parental phenotype. And so the man is normal vision, and the woman here is colorblind. And notice the genotype. Notice there's nothing on the Y because this is a sex link, X link, I should say. All right, and so being an X link, if you notice the uppercase C here, the uppercase C on the man, which represent normal, and then for the woman, she has recessive alleles, so that will be colorblind. So the colorblind is a recessive disease. All right, and so for the possible gamete, the male could produce X, which contains a normal allele, and also Y. The female, the woman can only produce X chromosome with the lowercase allele. So make no sense write it twice. All right. And so to complete the chart now, what you're gonna do is to put the gametes here. So this I put the gametes for the woman on the side and the male on the top. And so by pulling out this cross, notice I just put the uppercase in red to distinguish between the lower and the upper because sometimes it's hard to detect lowercase c and uppercase c unless they're really beside each other, right? And so the ones that are placed in red, those are the dominant allele. All right. And here you know, so the probability of colorblind daughter. And so the daughter would be here, and notice they're all uppercase C's, and so there's a 0% chance of a colorblind daughter. All right. And the point to note is that all the daughters will be carriers, which means they're hybrid or heterozygous. And all of the boys will be colorblind. So notice the boys here, they are lowercase C's. All right. Again, they're in blue, all right, to distinguish between upper and lowercase. All right. They didn't ask us all of this, but for understanding purpose, yeah, because no one's reviewing, we have to give some extra information here, right? Now for section B, question number 4 is a figure 5 is a diagram of the human respiratory system. Name the parts labeled A, B, C, D, E, and F. All right, and just to run through them quickly, we have A is the nasal cavity, B is the pharynx, which is also called the throat, C is the trachea, and remember that trachea contains rings of cartilage to prevent it from collapsing when you're swallowing food. And the reason because the esophagus is resting behind the trachea, and if you're swallowing food, it will press against the trachea, and the rings of cartilage will make it rigid and prevent it from collapsing whenever you're eating or swallowing food, as a matter of fact. All right, so D is the bronchus and is the left bronchus. The left part is not necessarily important. Once you say bronchus, you get your mark. But to put left to be specific, you can do that. We also have a left lung, and then F will be the diaphragm. All right, and so now let's see what they ask us here for part B. It says state 2 characteristics that the respiratory surface used for gaseous exchange in human beings has in common with the respiratory surface used in flowering plants. Explain how each characteristic makes the respiratory surface suitable for gaseous exchange. And again, it doesn't matter if it is human beings or plant, because respiratory surface, they do have certain things, well, pretty much everything in common. But I'm going to point out one thing that I don't list here. And so one of the major one is that thin. And the thinness of these surfaces is to allow gases to diffuse through easily. If it is very thick, then gas will take a longer time to travel, right? 2 must be moist. And the reason why it to be moist is that because the gases, they diffuse as mixtures, which we call solution, all right? And so diffusion of gases is easier when moist, all right? And then now we have large surface area. And large surface area is really to maximize the flow of gases, which increase the rate of gaseous exchange. So bigger surface area mean more gas can travel in or out of the structure itself, right? And then the next one is that it must be permeable. And being permeable, which means it's going to allow gases to move in and out of the structure. In other words, it will facilitate gaseous exchange. All right, the next one I didn't list right here because that one can be a little bit tricky. Well, it's not really tricky, but in terms of, for example, animals, and this is a difference actually, because animals will have a lot of blood vessels, right? So there must be something whether it be a fluid or blood and blood vessels around that structure to take away the gases. All right. So in some plants, you'll have fluid that are surrounding the cells or the structure. And in human beings, you have a lot of capillaries covering that structure. So if you notice the alveoli, for example, the alveoli, they are covered in a lot of capillaries. All right. And so let's jump into part C. For part C, it said advertisement for electronic cigarettes promise to supply nicotine to the body without harmful effects of cigarette smoke. These cigarettes are promoted as a healthier alternative to smoking regular cigarettes. So part 1 is to outline the effects of 3 named components of cigarette smoke other than nicotine on the human body, and just to point out that nicotine is addictive, but they say other than nicotine, so that's why I have that in red. And so one will be acetaldehyde, a matter of fact. And acetaldehyde, it is carcinogen, which means it can cause cancer. Matter of fact, let me just tell you, all of the things pretty much within cigarette smoke can cause cancer. And other effects will be that it irritates the respiratory tract and also the eyes. For acrolein is also carcinogenic. And also a mutagen, which means it can also cause changes to genetic materials as well. So it can lead to mutation. And we have irritation of nasal passage is another effect of that. We also have benzene, and benzene is a ring organic structure. All right. And so it is also carcinogenic and also affects the red blood cell and bone marrow as well. All right. And we have aromatic compounds, and there are different types of aromatic compounds found in cigarette smoke, and generally they are carcinogenic, and also they may lead to lung damage as well. All right. And you have carbon monoxide, and carbon monoxide can cause you oxygen to be deprived of oxygen, and so if you notice can lead to unconsciousness. I mean, well, if you're smoking cigarette, you won't have enough to really put you to a state of unconsciousness. But definitely carbon monoxide poisoning, if you know about it, it really decreases the oxygen uptake by the blood. And then you lose consciousness, and then you may eventually die. All right? So those are the general things. And of course, you may also have carbon that forms tar. I should list that one because the carbon compounds, they will form tar in the alveoli and then decrease the effectiveness of the alveoli as well. All right, so next question. I think I jumped over this too fast. Okay, yeah, I'm at the right place. All right, so part 2, let's make sure. All right, part 2 is outline 2 ways in which the use of nicotine in electronic cigarette may be harmful to the human body. And one, because of addiction, you become dependent on these thing, and when it said harmful to the body, being addictive to something or depending on something, then you will have psychological problems. For example, you can have mental issues. All right, because if you cannot get this thing, then it's also, even though it's not a biological, well, I won't say it's biological in a sense because it depends on the triggers, the biological triggers, and so you realize that you may have mental breakdown. Your mood swings. Social problems may arise from this. If you cannot get what you want, sometimes, it may lead to other negative impacts. Dizziness is one. And again, dizziness, as we mentioned, because of lack of oxygen flowing into different parts of the body, particularly your brain. All right. Also may cause increase in blood pressure and thus may lead to heart attack. It may affect the blood flow and also the clotting of blood as well. So there are a number of things that nicotine may cause in the body. All right. Let's jump to the next question now, question number 5, and we're almost there. It's a name 3 components of human blood that are important in protecting the body against pathogenic diseases and state the function of each. And so we have phagocyte, and phagocyte engulf and digest pathogens. And we have lymphocytes, and lymphocytes reduces antibodies that destroy pathogens. And we also have platelets. I know platelets work is like a precaution. As a platelets now will prevent the entry of pathogens, especially after they get a cut or injury. All right. Now let's go to part B. And part B is a long question. So let's go through this. All right. So in part B, it's a Marian, a 1-month-old baby, acquires immunity from her mother through breastfeeding. Marian's father accidentally punctures his finger with a nail but does not need a tetanus antiserum injection because he received a tetanus vaccine the previous year. Explain how the type of immunity that Marian acquired from breastfeeding differs from the type of immunity that her father acquired from the vaccine or from his vaccine. It's in your explanation, state the name of each type of immunity. And for Marian, her immunity is passive, and you're going to mention that. And how this works is that the mother produces antibodies and transfer them to Marian. And so her body does not produce the antibodies or have been exposed to the infection. All right, so she acquired it by passively. And a point to note for Marian's type of immunity is that immunity is gained without exposure to the infection or pathogen. All right, so it's a transfer of the defense mechanism from the mother to the child. And so for Marian's father, his immunity will be considered to be active, and the father was exposed to infection through vaccination because vaccination is an introduction of the pathogen or inactive form of the pathogen. Of course, now they're making different types of vaccine with RNA and so on, or protein based, which is a little bit different. All right, and so hence his body develops the antibodies needed to destroy the pathogen. And so his immunity is gained through exposure to the infection or pathogen. All right, so it builds up his immunity in terms of exposure. So his body will produce these antibodies and remember to attack quickly or remember how to produce these antibodies in case of infection. Now, for part C, it said the authorities prevent children who are not immunized from registering in public schools. It suggests two reasons why some parents may have a valid concern about immunizing their children, and two reasons why the authorities maintain their position and preventing the entry of unvaccinated children. And so for parents, there's so many things you can talk about right here, but once it's sensible and in line to what is happening here, then you will hear the mark. And so concern for parents so concern for parents here is that one, possible side effects or complications by taking vaccines, exposure to the pathogens. I mean, even the understanding of exposing to the pathogen. Once some parents hear that, hey, the child will be exposed to this pathogen, then they may have a concern, what if or what not, you know. And that is also lack of knowledge as well, because exposing to the pathogen will be a dead form or an inactive form. The next thing is that you're unable to tell if there will be any negative response. So since you're not sure of the response, you know, because really is a chance. And also another one could be age tolerance to vaccination. Is the child really to be exposed to this type of vaccine, or the child body can tolerate this type of vaccine? And for some parents too, they may have children that are sick or allergic to certain things. You may never know. All right, and so another concern could have been unknown future effects on development and also on the reproduction of the children. And the next one is that some parents maybe they are concerned about the components that are present within the vaccine. So this one's supposed to be unaware of unknown, or I can just say unknown components, right? Which mean they're unknown to the parents. So the persons who developing the vaccine, yes, they will know. All right, so they're not sure about the components present in the vaccine. All right, in terms of authorities now maintaining their position, is that there are a number of things right here we can talk about. One is that the authorities will say this will prevent rapid spread or people contracting diseases, and because they know that school involve a lot of communication and close contacts. So of course that will be like a melting pot to transmit the disease from person to person, and so they will stand their ground to reduce transmission of diseases. Two, is that less possible persons visiting health centers. And so if um, not going to school will kind of force people to get vaccinated. And so once it prevents people from going to, or students from going to school, then that kind of force a majority of the population to become vaccinated. And so that will reduce strain on the medical sector and also medical bills in the future. And also, if students are vaccinated as well, just in case you have an outbreak, you have less possible absenteeism. All right, so those are some basic reasons for both. Again, anything that is sensible and in line to what is making sense, they're sure going to get the marks. Now, for question number 6, last question, part A, is to describe how the process of mitosis results in the production of identical cells in asexual reproduction. And so I kind of do this in 2 ways. I want to kind of notice something here. For this, I put the steps or the phases, right? And also on the left, I put the number of chromosome, and I use an example number here. Just kind of give you a guide in terms of what will happen. All right. And this is not for human beings, because if it was human beings, then I use 46. So the number of chromosome over here, I just use 10. All right. That's just a random number. All right. So the first phase we'll have here is the interphase, right? And so the interphase is that you have DNA replication. And just to make a note that the chromosomes are not replicating, it's the DNA that replicates. And so because in the interphase, what you still have here is what we have chromatids. Point to note, if we're starting with 10 chromosome, that will be our 2N, and 2N means the full number of chromosome, which is a diploid cell. All right. And so in the prophase, what happened? The chromosomes condense and become visible. And so chromatids now turn into chromosomes. And here you still have the same number of chromosomes. All right. So it'll be 10 as well. So you still have your diploid number. And now in the metaphase, you still have the same number of chromosomes, because the only thing happening here is that the chromosomes that are now become visible, what they do is align in the middle of a cell along the metaphase plate. So that's the only thing happening here. And then now the next thing is that after the metaphase, we have the anaphase. All right, and in the anaphase, we have the sister chromatids separate and move to opposite sides of the cell and form individual chromosomes. And the reason why they form individual chromosomes is because the centromere is still intact. And it's the centromere that determines the number of chromosomes. And since the sister chromatids, they separate and form individual chromosomes, then what happened here is that the number of chromosomes double. So in the anaphase, you have a double number of chromosomes. You have twice the diploid number. That's why we have here 4N. Okay? And then now we have a telophase. And the telophase, the same number of chromosomes exist. The only thing that starts to happen here is that the cleavage furrow forms, initiating the division of a cytoplasm. The nuclear membranes will form and envelop each set of chromosomes. And a point to note, I just outline what is happening to the number of chromosomes, right? There's a number of different things happening, such as the spindle fiber forming, right? And the centriole forming and those things. And they move and how they pull the chromatids and so on. So those things you can mention. I just want to give you in terms of number of chromosomes what is happening here. All right, because looking at identical cells. Again, you can put in little details in terms of, again, as I said, the spindle fiber and so on. Now, after telophase, we have the cytokinesis. And cytokinesis, now what happened here is that the cytoplasm will divide and separate in the two nuclei, because in the telophase, we have two separate nuclei, right? Because the chromosomes are on opposite sides. And so once the cytoplasm divides and form two identical cells, they will have the same number of chromosomes as the parent cell. And so each cell will be 2N, which means 10 chromosomes each. Again, the number 10 that I use is just for example purposes. All right. Again, if you know if this is human being, then you have 46 chromosomes. So a quick point to note right here, or just as a summary, I just want to remember that in the mitosis, you have I, which is the interphase, and then you have PMAT, and then you have C. All right. So just to make a note of that. So that will be the summary. I, PMAT, C, the process of mitosis. All right. Now let's jump into the next part of this question. The next part of question, part B, is plants that reproduce sexually have some advantages compared to those that reproduce only asexually. Outline 2 advantages and 3 disadvantages of sexual reproduction. And so advantages of sexual reproduction is that it results in variation, which means you have an increase in biological diversity. All right. And next, and biodiversity, a matter of fact, also pointed out, is better because it increases the survival of other organisms and even the organism itself. And so here now, a second point here that the sexual reproduction can suppress unfavorable traits, because I mean, if you have an unfavorable trait and then a next plant with a favorable trait, then they come together. If a favorable trait is dominant, then it will suppress the recessive unfavorable trait. All right. And a third one here is that it is a slow process. And then if it's slow, then what it can do is to reduce the possibility of overpopulation. Now, a point to note, for technically, you cannot get these things wrong unless you state it a certain way. Because the truth is how you state it. Because an advantage can also be a disadvantage depending on how you state it. Here now, a better chance to adapt. All right. So again, sexual reproduction results in better adaptation, and it's also inched on part on the first option, which is increased variation. So more variation will lead to better adaptation. And so generally, sexual reproduction as an advantage can increase chance of survival because of these possible other options that I listed before. All right. Again, stating disadvantages and advantages is based on how you state them, because either one can use for the next. All right, so a disadvantage here for sexual reproduction is that it requires fusion of gametes. All right, and a point to note here is that gametes do not always meet. Fertilization is always by chance. All right, so there's only a chance that the male gamete may reach a female gamete. It's not a guarantee. All right, and also the gametes may not always be viable. Just to make a note of that. All right, and next one is that sexual reproduction takes time and energy. All right, and so population growth is slow. Again, this can also be an advantage if the population was at a stage of overpopulation. But if it's underpopulated and you need them to populate an area real quickly, then taking time and energy and it's slow, then definitely that will be a disadvantage, right? And so a point to note here is that because of this, you have slow recovery time after natural disasters. All right. And next one is that you have few offsprings compared to asexual. And then again, a point to note here why this is as a disadvantage is simple, that if the organism is a prey, all right, and this is in plants, so that's also fine, is a producer. And so if it's few offspring, then it can become extinct, especially if it is overexploited. All right. And so our next point is number 4, and this one I put some asterisk here to point out that depending on the organism, may need parental care. So some organism, and this is, since we're talking about plants, I wouldn't necessarily list this as I put some asterisk here, but just to make a notation of sexual reproduction. So depending on the type of organism, some organisms need parental care, and because of that now, the parent may not have time to reproduce again because they're taking care of the offspring. All right, so they take a longer time to go back into that mating season. Number 5 is that slow development of offspring. So sexual reproduction, offspring from sexual reproduction tend to develop slower than those that come from asexual reproduction. All right, and so we're on part C, and part C1 is that scientists have successfully cloned animal species. So there's 2 reasons why humans may choose to clone animals. And one is to maintain or increase population size. All right, so if population size is good, they can use to maintain it or to increase it if it's low. And 2 is to prevent extinction or of threatened or endangered species. They may clone animals as well that to help the organism reproduce if there is no suitable mate or if no mate is available. So for example, if you only have male animals, for example, you may want to clone them so you can have more of the species. If you have only female and no male, the similar thing could happen. Another one could be for research purposes to study development in that specific species. All right, and then and reason why this too, because if they're cloning the organism, they get it from the very tissue, and so they can see how the tissue start to form different layers and so on and become the organism. And six is to create more organisms that can be used to extract medicines. So if the particular animal that used to extract medicine, they can clone to have a lot of them in a short period of time, and so they can use that to produce medicines. So that will be for profit, right? And so outline two ethical issues related to the cloning of humans. And one of the first thing that we will always see when it come on to cloning is that people playing God, all right, or scientists playing God, all right. And so one, you could have moral consideration to human life and development, as well as people may say that if you're doing that, then you have no regards for human life, all right. And the big question here is, who are the parents of a clone? Again, as we know it, a clone offspring or human being only have one biological parent. And so that again could be issue in itself, not biologically, but social issues, which I list as number 5. Another question here is, what if the clone fails or have to live with complications? Because how would you justify that you generate a clone, and then what will happen here is that if the person start to grow and develop and now become conscious of life, and then have to suffer through probably kidney failures, kidney failure, liver failure, and other complications, how do you explain that to the person? That cannot be right. Another one here, as I mentioned earlier, is that the social development could be a next thing, how they immerse in society. And of course, if you're different and persons know about it, then that could lead to social ills such as bullying against that person. All right. And so those are just some of the basic things. Again, any other thing that's suitable and you list them, you get your marks for it. All right. And so I want to thank you for watching. I know we are pressing for time for examination, but I thank you for sacrificing your time. And I want to tell you that I wish you luck on the examination, and definitely we will talk soon. So good luck if you don't hear from me for this exam season again, but we'll definitely talk. All right. So keep studying, keep working hard. All right. Take care.